My name's Adrian Kincaid. I'm the CEO of MIP Diagnostics. Been with the business now for 4.5 years. And in that time, really seen them come from a university spin out with no staff, no business plan, nothing, to what I think now is a business that's really prime for expansion, and hence going for this deck is all about the Series B. It's a bit of a work in progress, as these things always are. So if people have views or input and think we should go for more or less or that sort of thing, then if it's your money, we'll certainly talk about it. So if I can just move on. In summary, for this plan, we need an investment of 4.5 million. Given where we're at and the pipeline, we justify a valuation of 15 million. Things really are moving really very rapidly. Far too much text on this slide, but just rapidly running through. The leadership team is all in place. Everything is working very well. The revenues that we earn from our customer service work is starting to come in. Well, it is increasingly coming in. And we have a pipeline of license deals for recurring revenues on getting our MIPs into finished products and getting those products to market. We do not take the risk of developing a diagnostic, for example. We do not run the clinical trial. We appreciate that they are very expensive to do. We're in fact part of that cost. So the business is very much de-risked in that sense. But I can take you through some of the examples there. The technology that we have is a platform technology. It has a great many different applications. Our focus is very much on the IVD market, but the revenues that we've earned to date have been offering access to the platform technology on a custom service or contract development basis. And if our customers come from different fields that we hadn't thought of, but will pay us to do the work, then we also get to do what we call paid learning. And we can look at things in fields that we wouldn't otherwise address. So just to put some flesh on that bone, one of our customers is a multinational oil company, and they want to measure analytes in crude oil at 100 to 120 degrees centigrade and under tremendous pressure, as well as being organic phase, of course, and a filthy sample. I'll take you through some of the ways in which our technology is so different from other affinity technologies out there that we're able to address, or we hope we're able to address those markets. It's a research project at this stage. So the MIPs are totally different from other affinity ligands that are out there. But the easiest way of thinking about them is a polymer antibody or a plastic antibody. They're as robust as plastics are. So as you can see on the right-hand slide, please don't read all the words, they can have a tremendously long shelf life. The calculations suggest that that could be in excess of 10 years. Of course, we don't necessarily need our customers to advertise that if they don't want to. But it means that the polymer we can make, we can autoclave them. We can autoclave them, we can sterilize them, they're not biodegradable, we do not need cold chain storage. Shipment supply chains become very much simplified, very much cheaper for our partners to deal with, all because the technology that we have is these polymer MIPs rather than antibodies. We can work at very high pressures, as I mentioned earlier with the oil. And the sensitivity that we've developed for our flagship ones, just to show how they can do, they are better than the best antibodies out there at the moment. Our flagship is cardiac troponin I. People have been making antibodies to that for the last 20, 30 years. So those antibodies really are the best that you're going to get from antibodies. It took us 5 months to get equivalence with those, and yet we can autoclave them. We're continuing to do the development work, and we expect to exceed the sensitivity of the antibody-based assays for troponin in a really very short period of time. The business has worked through the business plan. We set out last time to raise 1.5 million to start doing our internal research, such as the cardiac troponin MIP. We've delivered on that. Last time we were actually oversubscribed. We got 1.7 million. And the revenues from the custom service business, which is not the underlying true value of the business, but revenues that we can recycle, is growing in excess of 200% CAGR. This is the technology then, and if I can quickly take you around in a sort of clockwise fashion, the coloured ribbon there is our template molecule. That's the bioanalyte. That's the target that we're going to be making a MIP to. You can see on that zigzag line, it's immobilized onto a solid support. So that's nothing more than glass beads. These are packed into a column-like format. And once in that column-like format, we can add those little molecules down at the bottom of the slide, sort of in the center there. There are monomers. Now, these monomers are equivalent, if you like, of the amino acids which make up proteins, such as antibodies. You'll be familiar, I'm sure, that there's 20 amino acids. We have a lot more chemical diversity. We can cover all of the side chains of amino acids, and we can do a lot more besides, because there's about 5,000 different monomers in the literature already. Monomers consist of a functional group which we hope will do the binding, and nothing more than just a carbon-carbon double bond finishing CH2 that we can polymerize. What we do is we introduce these monomers, a selection of them, typically about 7 at a time, 7 different species, in with the target, which is mobilized onto the solid support in that column, the affinity column. Each individual monomer can then find its lowest energy state, which is its optimal binding position. They self-assemble into their optimal binding positions. Each interaction is fairly weak. It could be just a hydrogen bond. So what we want to do is make them all work together as a single entity, and that's why we polymerize them in situ. So we start forming the green ribbon there to make the polymer. We grow that green ribbon until there's enough bulk to hold each of those binding monomers around the template molecule, and the green blob sort of on the bottom side there contains typically 1 binding site per nanoparticle. We stop the polymerization at that point. The idea of doing that is because we want discrete nanoparticles which we can then separate according to their affinity. So because we made it on an affinity column in the first instance, we just wash away the material we don't want. Keep the material we do want. We may even do a little bit of extra chemistry there because the binding site is occupied and therefore protected, before we then harvest and collect the nanomips that we need. We control all of the chemistry involved, so we can make them fluorescent, hydrophobic, hydrophilic, positively charged, negatively charged, pegylate them, biotinylate them. What would you like? Any particular color? Really do have that much control. We control the size of the particles as well. So we can make them as big as 300 nanometers, which means that we can make them big enough to bind an entire virus particle, or we can make them really small to get maximum packing density. In fact, if you take it down to the extreme, we can make them around 1 to 1.5 kilodaltons inside, which is starting to get quite drug-like, actually, and arguable as to whether they're really polymers at all. So the technology is remarkable, it's remarkably robust, it's remarkably versatile, and the affinities, of course, are what we want them to be because we control the wash step and the harvest step. Finally, if you do the same things in the same way in a chemistry environment, which is what this is, you get the same results. You do not have the variation that is inherent within biological systems. So we can make another batch, possibly 10 years' time, whatever. If we've got the same materials under the same conditions, we can expect the same results. So as I said earlier, our focus is on IVD and making MIPs to compete with antibodies or even improvements over antibodies. On core areas, we can enable testing in the field. We work with partners to get those products to market. We make the MIP, that's our specialisation, but we can work with a large number of partners. So there's a great many markets to go after. MIPs are really totally different from any other affinity reagent that's out there. There are other affinity reagents, but MIPs are certainly the most robust. And this also engenders in our customers, which you can see down on the bottom right, a real unique sort of collaboration. We offer something very, very different. They appreciate that, and they work really hard with us to generate MIPs that are going to meet their needs. We can tailor them, we can put in chemistry to make it work for them. They appreciate that, so they tell us more about what they need. And the more you understand, the better job you can do and the better product that you can release. So in terms of the business, we're ahead of schedule in terms of where we thought we would be. Recurring revenues is key. Getting these products licensed and out into somebody else's product is where we need to go to. I'm afraid this slide is now out of date, so there's 3 little green symbols meaning heads of terms signed. Sorry, 4 on there. The 5th one has actually turned green now, and we've got that one signed as well. So these are routes through to recurring revenues. Most of the numbers that underpin the values down in the licensing pipeline are actually coming from our customers and taking what they've said rather than our assessments of what we think they should sell. So hopefully that adds more weight and validity to what we can do. So in summary, we're looking to raise about 4.5 million against a valuation of 15 million. Other investors are in place. Mercia spotted the technology even before I joined the business. Calculus came in at the last round, and we've got various high net worths as well. The Series B is all about scale up. We've taken the risk out of the business now. We've got examples that show the advantages, show that we can do better than antibodies at their own game, even using epitopes which are optimized for antibodies. And we offer robustness that is unparalleled in the field. So at that point, we've got another couple of minutes. I'll stop and take any questions if anyone's got any.